Anatomy and physiology. So the last thing to talk about with lymphatics and immunity before we can move on to the respiratory system is antibodies. So we were talking last time about B cells, and one of the things that they do is differentiate the plasma cells and release antibodies. Antibodies are proteins. They are actually these Y-shaped proteins, which mean they have certain parts of them that are constant. So if you're looking down here, these ones right here, this red region is the constant part of these. And then they have these variable regions, which are shown in the pink, green, and blue. These variable regions are coded for by a few different sets of genes called V, D, and J ones there. And they are what give us a number of these different specificities, a lot like we talked about with T cell receptors and the fact that you have not a ton of genes, but different classes of genes where each class of genes is doing that can get you lots and lots of combinations without the use of a ton of different genes. So they talk about a lot of these fragments and other stuff like that. The main thing I need you to know is that they are Y-shaped proteins, and then these little points on the end here, this is where the antigen binds. So on any particular just single antibody like this, you're going to be capable of binding two different antigens or epitopes with that particular antibody. And this allows the antibodies to do a few different things. So in terms of what antibodies can do, we got a little list here, and I mean, to me, the first couple are kind of similar. Neutralization is just very simply, if we have antibodies present and we can stop the particular pathogen or a toxin from getting to its target, we neutralize it. We stop its ability to cause disease. So if we can release a bunch of antibodies and it can bind this up so it can't get to anything, that is a way to prevent this. This is kind of the idea if you were to prophylactically get antibodies against something, having antibodies injected into you, they can bind something up so it can't get to the target site. So example of this one we've already talked about was RhoGAM and having the mother get antibodies against the Rh factor. We are neutralizing that Rh antigen from getting to the specific T and B lymphocytes so we don't have an immune response. Another thing this can do is it prevent cells from really being able to move around or adhere to any areas. So antibodies, for example, can bind up bacteria, flagella, and allow them to not be able to get around, or can bind up their adherence molecules that don't let them actually attach anywhere. So that's a couple of things. The other one on this one here is it talks about is agglutinating or precipitating. Remember, agglutination was the antibody-antigen clumping response. The main thing is phagocytes. I always kind of talk about phagocytes being a lot like Pac-Man, like old school video game here, but everybody knows Pac-Man a little bit. If you had the ghosts where you could link all the ghosts together, it'd be much easier to eat all 4 ghosts. That's kind of what happens with agglutination and precipitation. In the case of the picture down there, you have these antibodies binding up a bunch of bacteria, so now a phagocyte can come through and just glom them all up versus trying to track them down one at a time. Another one here we've already talked about a little bit, but this idea of enhancing phagocytosis. If you have antibodies bind up something, bind to this particular thing here, you can trigger a phagocyte to eat it. This is called opsonization. Another thing that it can do is this is one of the classical ways that the complement pathway that we talked about is nonspecific, can be triggered by a specific immune response. So we can activate complements by antibodies binding to their particular antigen, and the things that come along with that complement activation, inflammatory response, opsonization, and lysis of these cells by those complexes that are made by the complement proteins. That is another one here. And then the last one here, antibody-dependent cellular cytotoxicity. This is targeting specifically natural killer cells, which are a non-specific defense to attack specific cells. These are the number of different things that antibodies can do. The other thing that is interesting about the antibodies is there is a number of different classes of antibodies. We talk about antibodies as being immunoglobulins, which if we kind of talk about the different classes, that gives you the little I, little g, so the Ig, whatever. We're going to see that there's actually 5 different classes of antibodies. We have IgGs, IgAs, IgMs, IgDs, and IgEs. Again, each of these is a little bit different in terms of what's going on with them. There's a few things that I think are key on each of them that I will point out. IgMs, the main thing with this one and the biggest deal with this one is it is the antibody of first response. So anytime, the first time you're exposed to an antigen, the first antibody you're going to make is IgM. It is always going to be that first one. It's not so worried about the percentages in there. It is 5 of those little Y-shaped proteins connected together, which makes it what's called a pentamer. The other thing that's interesting about this one is it's the only one the fetus can make before birth. So kind of interesting on that one. IgGs to me are kind of end up being the most important one here in that they are the most common one in circulation. So if you look at all the immunoglobulins that are in circulation in a person at any particular time, 80 to 85% of them are going to be IgGs. A few other things that are interesting about this one. This is the one that the B cells, once they've been differentiated, you're always going to make some level of these antibodies. So a lot of times we refer to this as that antibody of memory. It's a lot of times the fact that these antibodies are always in circulation. You can actually do titers of the blood for these ones and look at levels of immunity, things along those lines. The other thing that's interesting in this one, this is the one that's made by the mother, and it's the only antibody that can actually cross the placenta and go into the fetus. So during the Rh antigen issues we talked about, that was a bad thing, but most of the time that's a good thing. Anything that the mother is immune to, what that means is because these IgGs can cross the placenta and they're the most common type, if the mother is, let's say, immune to measles, when that infant is born, until those antibodies clear the system, so is the infant. So that's kind of a neat thing with that one. Then the other one is, the next kind of one that's probably one of the more common ones is the IgAs. These ones are found in a lot of different body secretions or glandular secretions. So things like breast milk, mucus, tears, saliva. This one's actually a dimer, 2 of them hooked together. Again, another way, mother to infant, that this can be passed along by the mother breastfeeding the child. The child can actually take in these IgA antibodies that can also confer a little bit of immunity towards certain different pathogens. The IgDs, these ones not nearly as big a deal. These ones are part of those B cell maturation process. These are found in the membranes of the B lymphocytes, the IgD ones here. And then finally IgEs. These ones really are only active in allergic reactions. Again, not something I'm going to go into. Again, this is an immunology thing. If you want to really learn about hypersensitivities and everything that goes along with that, I would say take a course like that. We just don't have time to delve that deep into it in A&P. So this is kind of, again, showing you the different ones here. I don't want you to know the molecular weights or anything else like that. I'm not even worried that you know the levels here. To me, knowing that IgGs are the most common, yes. But you can see the pentamer structure. You can see the dimer structure. The other thing this one does here is it talks about the properties and functions. To me, that would be the part on this table I would take a look at, which helps you understand what each of these different ones are doing. If you want to remember them in a particular way, you can remember GAMED, G-A-M-E-D, is the easy word to remember that kind of has all of them in there. When we've talked about this already, when it's getting in there, only the appropriate antibody is going to bind to these particular ones here. Those cells that are making these plasma ones there, without continued stimulation, a lot of these cells undergo apoptosis. So after we've clonally expanded these lymphocytes, they will kind of tone back down. So that's where we'll get that strong immune response, and then it will tone back down once you've kind of fought off the particular pathogen. What's kind of interesting with the antibodies, and this is one of the ways you can see that primary versus secondary response really well, is actually looking at some of these antibody levels or titers and seeing what goes on with them. So the primary response would be the first time somebody sees that, and looking at what happens with the antibodies, after that. It's kind of interesting. And what we're going to do on the next slide here is compare that to a secondary response. The second time it comes into contact with an antigen, what's different? So you can see on this figure right here, first exposure to an antibody, first thing you notice, you have this latent period. What's going on with the latent period? Well, that is that clonal expansion, the differentiation, those things that we talked about in those primary responses where you have to get that clonal expansion memory cell set aside, everything else like that. What we are then going to see is the purple is kind of the total level of antibodies, red is the IgMs, blue is the IgGs. What you notice, there's a latent period, then the first antibody that's going to be made is the IgM. After that, you see the IgGs going up and you get a particular level of response. If we look at the secondary time, you see this, let's say this is a year later, you get exposed to the antigen again. First thing you notice, what's missing is the latent period. There's nothing there. The other thing you notice is while the IgMs come up at about the same level in the same time course, we see a much stronger and much faster response with the IgGs titer going way up in there. So what you notice on this one, the secondary response, we get a much quicker response and a much more robust or stronger response in terms of antibody levels. What this means a lot of times is you don't recognize you've been exposed because you don't get sick. So really the last stuff to talk about here is just a little bit about the different ways we get immunity against something. So you can see there's a number of these ones here. So you can see all of them have acquired in there, all of them have immunity. So we have what to kind of differentiate between is what is the difference between active and passive immunity, what is the difference between natural and artificial ones here. And then we'll talk a little bit about just vaccines and immunity. And again, a lot of this is going to come into stuff that we're dealing with right now with the COVID-19. I'll make some points on that at the end here. So the main thing here, when we're talking about acquired immunities, active versus passive is really just getting into where do the antibodies come from. Anything that is an active immunity means you've had an immune response, you're generating your own antibodies. Anything that's passive means you're receiving antibodies from another source, you haven't had the immune response, you're not generating your own. So that is showing you the active right here. So what it means then is if we start talking about active immunity, we then have to differentiate really how was the exposure to the antigen. Was it natural or was it artificial? How are you naturally exposed to an antigen? You get sick, you get better. That's how. You get the cold, you get better from the cold, you have antibodies against it now. You had your own immune response. That is the natural way. What is the artificial way? We inject some type of non-infectious antigen into you that triggers you to make antibodies against the more live version of this. So this is what vaccines are. This is what immunizations are. We have either part of that or killed one or sometimes a weakened live virus, something along those lines. We put that into the body then has an immune response against it and makes its own antibodies against it. That is what they're working on with the COVID-19, trying to come up with a vaccine against that. So if we go the other direction now and talk about passive immunity, we're receiving ready-made antibodies. What this means is you didn't have an immune response. The other thing this means is it's not a long-term type of immunity because antibodies in circulation, if you didn't make them and you're not continuing to make them, they last at most about 6 months in circulation. So how do we get naturally made antibodies? It's mother to infant. It's that vertical transmission of it. So either from the mother passing them across the placenta to the fetus would be one type, or the mother through the breast milk to that infant would be the other way. But again, maternal antibodies provide some immunity for a while, but after about 6 to 8 months, those are out of the system. And then you start seeing these infants get sick with a lot of different things. How do we artificially get antibodies, pre-made antibodies? This is one of the things they're talking about trying to treat with COVID right now. Getting these patients that are convalescent patients, ones that have recovered and have antibodies against it, if they take their plasma and purify out those antibodies, if you inject those antibodies into somebody else, it can confer a short-term immunity. It can help them fight it off. And that is how you would do passive artificial. And again, this kind of ties it all together here. Active immunity, making your own antibodies. Again, either through following an illness would be the natural way. Through immunizations would be the artificial way. If we talk about passive immunity, it's ready-made antibodies from another source, either across the placenta or through breast milk are the natural ways. Artificially, by the lady smiling here, is she's getting an injection in her rear end. Usually you don't do that when you get a shot, but anyways, you're getting ready-made antibodies injected into you or through an IV. And again, this just shows all of them. So one thing I want to talk about with vaccines is some are different than others. Generally, the closer it is to the live version, usually the stronger or longer lasting immunity that there's going to be. So live viruses a lot of times tend to do a little bit better than killed ones or ones that are just pieces or toxoids. Some of these give you for a single dose, other ones you have to do multiple times. So live vaccines, usually a single dose is good enough because the microbe multiplies in the body a little bit but doesn't cause sickness, and the immune system really strongly responds to that. Where a lot of times these attenuated other ones here that are a little bit less, these live ones, if somebody's immunocompromised, they can be an issue. If somebody's pregnant, they can be an issue. Some examples of this are yellow fever, measles, mumps, rubella, or attenuated vaccines. Inactivated ones, sometimes these are the whole thing killed off. Other times it's just fragments. And again, immunogenic means they trigger a response, but not pathogenic means that they can't replicate at all. Things like tetanus is a toxoid. You would need continual boosters of that to keep your antibody levels up to avoid that one. So that's one that we see is not quite as immunogenic, and you need boosters every so often in order to have that happen. This is what we're trying to reach with the COVID-19, the main problem with it is nobody was immune. And because of that, there was no herd immunity. What that means is everybody that's exposed to it is capable of getting sick from it. And that's why it spread like it has and why it's done this logarithmic increase of the populations, and they go up and then go down after the fact. With the stuff that we're able to vaccinate against, things like measles, mumps, and rubella, we don't see a lot of outbreaks of that. Why? Because when 90% of the population is immunized against this, it's very difficult for that particular pathogen to find somebody that's susceptible to pass to. So what we're trying to do right now is avoid contact, because right now everybody is on this end of the spectrum where there's very few people that are immune to it. Because of that, it can spread very easily within the population. So we are trying to reduce the number of people that are around one another until we can get that vaccine and do something like this on this end of things. So the idea that once you get enough of the population immune against something, that gives you what is called the herd immunity.